डाउट्स में अटकते,
सब्जेक्ट्स में लटकते, कब तक हम रहेंगे सर पटकते।
अब वेदांतु देगा प्रॉब्लम से छुटकारा।
समझ आएगा तो मजा आएगा, मजा आएगा तो समझ आएगा। बढ़िया टीचर्स प्रॉब्लम्स चुटकी में सुलझाएंगे, डाउट सारी जिंदगी से
Raj, I got your doubt. This is your answer.
समझ आएगा तो मजा आएगा, मजा आएगा तो
Vedantu. Learn live online. To attend a free live class, download the app now.
Hey students, good evening, namaste and welcome to another session of a brand new sub-series of our existing ICSE Class 10 Biology series. Yes, hi Himanshu, Rajdeep, Ayush, Fury Fist, Shreya, Rajdeep, Devender, Jai. Yes, I can see a lot of my regular students also out here. Thanks a lot. Yes, hi Hasini, Anushka, thank you very much for joining me today. It feels so good to be doing a live session after quite a break. Yes, good evening, Shreya. I hope all of you are doing great. Your pre-boards must be happening now. For some of you, it must be about to start. For some of you, the second pre-boards must be about to begin. So whatever situation you are in, it doesn't matter. You are still almost a couple of months away from your board exams, which ultimately matters the most. And that is why we have come up with the brand new diagram series, as you can see it, which will run for the next 5 sessions, including today. Starting today, it will be running for the next 5 sessions, so that we can discuss all the most important diagram based questions. Okay, all right, so just in case you are attending my session for the very first time, let me not forget to introduce myself. I'm Ambika, Master Teacher of Biology at Vedantu, and let us get started with our diagram series, session number 1. Yes, hi Varshit. Okay, new student Reshma. Never mind. I am pretty sure there would be a mix of CBSE and ICSE children as usual in this case. Again, yes, Bhavya, I am getting better. I am gradually getting better. I've been advised not to raise my voice much. Unfortunately, that is a very, very difficult thing for me. As such, I'm very talkative also, and my volume is also by default a little on the louder side. And plus, professionally, I'm a teacher. So everything, let's see how it goes. Let's take it as it comes. Okay. Yes, and as you know, we have been doing a lot in this series, in the ICSE series, especially for biology. You know that we are done discussing and covering your entire syllabus of biology. Thank you all, my dear children, for your love and support and for being there to motivate me and keep me working hard for you and have a lot of interactive sessions with you all. We launched in the month of October, if you remember, and here I have some very, very special news for you. You can win a micro course of your choice at just ₹1 rupee. Okay, and here is how. Take the one class challenge. This is stepwise of what you have to do. Download the Vedantu app, the Vedantu learning app. Select your grade and choose any free masterclass of your choice. All you need to do is just share the screenshot of the masterclass that you're watching and the link of the micro course of your choice to this email ID. Make a note of that, youtube@vedantu.com, and then you would get a confirmation mail with a coupon code, and use that coupon code to get your choice of micro course at ₹1. Okay, yes, let the learning begin, and here we go. Keep using our Vedantu learning app. Thank you very much for all your support until now, and do keep supporting us. We are also always here to keep working hard for you so that you can just focus on your studies. Okay? Yes. And the Telegram group, as you are aware, many of you are already part of the Telegram group. Those of you who are not and would like to be part of it, please do please do hit the link which is given in the description box below and join us in a lot of amazing discussions which will help you join the respective group, and you can get all your academic related queries all solved from expert teachers and expert mentors out there. Okay, and as I always tell you, do not forget to hit the like button, share it with all your friends, especially the diagram series children. Please share it with all your ICSE friends, and do remember to subscribe to Vedantu 9th and 10th if you haven't done it yet. Yes, hi Mansi. You're welcome, Pratosh. Okay, yes. Before I read some of your names, it's just scrolling up, so do not feel bad at all if I'm not reading some of your names. That doesn't matter anyway. For me, all that matters is your participation, your active participation, so that we can get the best out of the session. Okay, so let us discuss, as I promised in yesterday's session, let us discuss the homework answers of our last two sessions, which were both on sense organs, right? Remember, the first day I gave you a question based on the ear, the human ear. Okay, and here are the answers I got for it. Hasini, who sends emails and puts comments from the ID Rajnikant Vadapalli, you must have wondered how I know Hasini. She normally stays in touch with me. I know you are part of the session also, right? Hasini, I saw your name somewhere. Yes. Okay. So Hasini has given me the answer first, and then everyone else. Ayush Sharma, again, after a long time, I'm hearing from you, and then Arul Das, Archana F. Smith, that right? So Hasini says it's caused because of motion of vehicles, which causes disturbance in the inner ear. For some people, they get a vomiting sensation also. I think I did tell you about my own case wherein motion sickness, especially if I'm traveling in an air-conditioned car sitting on the back seat, many times I do experience this motion sickness and I do get this nauseous feeling, vomiting sensation also. Now Ayush says, yes, due to confusion or wrong interpretation caused by signals from the inner ear and brain, basically. Yes, okay, yeah, that's about it. I thought the answer is continuing, and that explains the answer. Everyone, thank you very much for giving me the answer, explaining it crisp and short. And here we go. Archana had asked me, as you can see here. Yes. Archana says, I have a doubt, what is the function of round window? Okay, and I had also told in yesterday's session, I had mentioned this also specifically, Archana, that I will be explaining this in today's session. So here we go, and thanks for reminding me once again, Archana. So this is the outer ear, the middle ear and the inner ear. As you can see, there are two membrane bound structures which are called the oval window and the round window, right? Oval window, we have seen the function of that, and the round window also, in fact, it supports it when there is pressure going in to the inner ear from the middle ear through the oval window. In fact, there is pressure going out of the ear through the round window in the opposite direction. And that is how it's able to bring about that balance without causing too much of pressure disturbance. In Archana, that solves your doubt, and a lot of others who may have had that question in mind. Thanks a lot once again for asking. Yes, again, now this is the homework question which I gave you yesterday, and the answer for that particular question came from Shagun. Thank you very much, Shagun, for giving me the answer to that. This is due to the reason that in dark room the pupil is large, so when we come out in bright light, a large amount of light enters our eyes and we feel the glare. Okay, but after some time the pupil contracts and we are able to see clearly as less light enter our eyes now. Okay, now why did I choose to put all of these? It's basically because I had asked you two questions, but I was disappointed to see that there were very few answers that came for the second question. Yes, of course, all the diagrams will be easy for you, Soumya, and that too if you are in ICSE, definitely you are lucky, because unlike CBSE, which asks students to draw a lot of complex diagrams, ICSE doesn't usually give students that trouble. It's usually diagram based questions more than learning to draw diagrams. Okay. Reshma, it is more for ICSE children, but if you like to get more insights into the topics, you can certainly be part of the session. Okay? Yes. All right. So here we go, and Shagun also says iodopsin regenerates and rhodopsin degenerates, due to which we are able to see the objects. And I had shown you a picture as the second homework question. What color do you see? And I was a little disappointed, as I was saying, that only a few answers came. Shagun says the color of the dress is blue and black. Jai says the color of the dress is blue and green. I did not put it again here. Those of you, yes, Jai, blue and green. I have read out your answer, okay? Those of you who have seen that picture or who have not seen that picture, please do go back to my yesterday's session on sense organs part 2. Tell me what color do you see? It's a very, very interesting question to be discussing. I didn't put the picture because that may end up taking the rest of our time for today's session, okay? Yes, these are also answers that I received for yesterday's question. Ayush Srivastava and Jai, again, you have given the answer for this question also. Ayush also says, everyone says the same thing. Brightly lit room to a dark room. Yes, this is called dark adaptation. And basically, I think I've explained this to you when we discussed the specimen paper, mainly because which is today we shall be discussing diagram based questions from 3 of your major chapters, which I have broken down into 4, which means structure of chromosome I have put as a separate chapter. If you recall, and if you have been part of my ICSE series from the launch, you would remember I taught that as a separate chapter, because in the old ICSE textbook it is actually as a separate chapter. Okay, so whatever it is, what matters is all the fact that you get familiar with how to answer such questions. Okay, it's definitely impossible to discuss every possible question, because that would probably take us another 2 or 3 months, and that doesn't help, because we need to keep giving you a lot of more interesting stuff also, right? So we will continue this for the next 5 sessions. Okay, yes, Yash. So all right, okay. So the first chapter that I have chosen is cell cycle and division. As I said, we will be discussing one question from each of these, and you will get a better idea, better insight into how to handle diagram based questions once we end today's session. With the next remaining 4 sessions, you will certainly be able to boost your confidence better. We will be taking 2 or 3 chapters diagram based questions together in one session. Okay. Yes, today also I have a homework question for you, so do not leave the session. At the very end, let us see who's going to give us the best answer. Okay? Yes, here is the first question. Study the diagram given below, which represents a stage during mitotic cell division, and answer the questions that follow. Okay? Yes, Reshma, probably, yeah, in the diagram series, you can expect me to be telling you the most important types of diagram based questions. Now, it's important to note, children, that whatever I have put here is only as a sample. Consider it as a sample. These can be asked in many different ways. Cell cycle and cell division, for example, because of the number of stages it has in the cell cycle and the process of cell division, any stage can come. Yes, who's going to tell me the answer? Yes, Kanu Kumar, that was the first answer I saw. Unfortunately, yes, I was busy talking to all of you. Telophase, yes, as all of you say, telophase is the answer for this. Telophase or telophase, as some would call it. Okay, so let's look at the next question. Name the part numbered 1. This is 1, okay, and this is 2. This is 2. This is 1. Okay. Yes. No, I did not ask you for the reason. I'm asking you to label it. 1 is chromosomes, okay? Archie says 1 is chromosome. Vivek says chromatid. It is in fact chromosome, okay? Not the chromatid, because it's telophase. Chromosome will be 1. 2 is very good. Archie says nuclear membrane. So yes, 1 is the chromosome, and 2 is nuclear membrane. Okay? Yes. Very good. All right. Question number 3. What is the technical term for the division of nucleus? Yes, children. Now who's going to tell me the answer? Technical term for the division of nucleus. How many of you remember this? No problem, Jai. Karyokinesis. Yes. Dranzer Giggs the first says karyokinesis. Okay. Very good. Karyon is the technical term for nucleus. Karyokinesis is the technical term for nuclear division. No, it's not karyocytosis, it's karyokinesis. Division is karyokinesis. Okay? Take care of the spelling, Aryan. Okay, it's K. All right. The next one. This is a little more trickier. Mention the stage that comes before the stage shown in the diagram and draw a neat label diagram of the stage mentioned. Yes, children, what comes before telophase? Yes, Dranzer Giggs the first again says anaphase. Great job. Yes. Shagun also says it's anaphase. What does it look like? Of course, you can't draw and show me the image. So I have just put it out there for your reference. It is definitely anaphase. Now, how do you remember the sequence, children, of the stages of mitosis? Interphase, prophase, metaphase, anaphase, telophase. Telophase is what was given in the question. What comes before that is anaphase. Ana means opposite. Basically, the sister chromatids will be at the opposite poles. Okay, so this is just there as a reference diagram. So you can certainly draw it better than I'm drawing it on this board. You must draw the centrioles from which the spindle fibers are coming, but of course much more than that, it is the chromatids that you should be drawing. You can draw 3 or 4 of them on this side also. Chromatids, okay, and very, very important part would be the spindle fibers. Yes. Okay, spindle fibers should be attached to the center part, which is the centromere. Yes. Okay, children. Of course, this is only a rough diagram, okay? But one more important thing, when you are asked to draw a diagram, you shouldn't be leaving it just as the diagram. Of course, you must mention the stage that comes before telophase is anaphase. And this can be represented as follows. Draw a diagram like this and you have to label. You have to label this as the chromatids, or you can label 2 of them as the chromatids, and you can label this as the spindle fiber. You can label this as the centromere. Okay? All right, is that clear? There is no nucleus anyway because it's anaphase. Okay, these are the most important labels. All right? Yes. All right, now let's go ahead. The last sub-question for this one, which is the cell division that results in half the number of chromosomes in daughter cells. Hi, Vaibhav. Yes. Yes, Nirupam, your doubt is telophase and cytokinesis, right? Telophase is followed by cytokinesis. Telophase is a stage where the cytoplasm is still joined. If you look at the diagram, cytoplasm is still not completely separated. Cytokinesis is splitting of the cytoplasm also. Okay? Meiosis, great job. Everyone has been answering it correctly. Meiosis is the answer for the last question. What results in half the number of chromosomes? Equational division is mitosis. Reduction division is meiosis. Okay, all good. Great job, guys. Thank you for the participation. Next question is from the topic structure of chromosome. Okay, structure of chromosome, what sort of questions can you expect? But children, before I go on to that, let me tell you one very important thing. Now, as I told you, this is just one example of something that can come from cell cycle and cell division. Any of the stages of cell cycle in general, or mitosis specifically, or meiosis specifically, can come for your exam, and questions based on that can come. As you can see, everything needn't be direct. Unless and until you know the stages properly, the features of every stage properly, and you know how to label it and what each part is called, you will not be able to score full marks for such questions, okay, from this chapter. And definitely you can easily score 5 marks completely with a little bit of extra care. All right? Yes, so coming to the question on structure of chromosome. Figure A and B show two types of chromosomes. Identify them and write the difference. All right, tell me. A and B. What type of chromosome is A? What type of chromosome is B? Yes. Everyone's just blank and thinking. A is X No, I am not talking about that. Okay. Yes, I see the answer now. Shreya says metacentric, and that should be giving you some hint. Which one is metacentric? Saurav says telocentric. No. Jai, it's not autosome and sex chromosome. We are not talking about that. Shagun says A is metacentric. No. Metacentric. Meta. That should give you a hint. Meta means middle. Okay? Whichever chromosome has a centromere at the center. Yes, Kirtana, I do remember you. Whichever chromosome has a centromere at the center, as you can see, dividing it into two equal sized arms, you call that metacentric. And when it is towards the tip, not exactly at the tip, but towards the tip, it is called acrocentric. Nobody told me that answer. Metacentric, a lot of you did say. Okay. Yes, Arjun. Okay. All right. So, acrocentric and metacentric. I remember explaining this in the chapter structure of chromosome. So, those of you who have watched those videos will definitely understand the answer. This part where the sister chromatids attach is what we call the centromere. How would you explain it? When the centromere is at the middle, it is metacentric. When it is towards the tip, it is acrocentric. But when it is extremely at the tip, it is telocentric, which is an answer that many of you gave. Okay? All right. And if it is slightly away from the middle, there is a name for that also, which is called submetacentric. All right? Fine. So that's about a question from structure of chromosomes. Now, one of the most important and my favorite topic, which is genetics. Now everyone is talking about the song. Okay, let's talk about that a little later. Focus on the session. Okay. Yes, there are a lot from CBSE board also who always attend ICSE classes. Mendel, yes Soumya, Mendel. Let's look at a very interesting question, a problem-based question from, is my name is Ambika. Yes, Seema. Okay. Okay, so this is the question. In a homozygous pea plant, axial flowers are dominant over terminal flowers. That's the condition that's given to you. Let's see who's going to answer first. Okay, so pay careful attention, children. Axial flowers are dominant over terminal flowers. What is the phenotype and genotype of the F1 generation if a plant bearing pure axial flowers is crossed with a plant bearing pure terminal flowers? Okay, so let us try to work this out. If you can tell me the answer without working it out, even great, even greater, I mean even better, but I would like to explain it for the benefit of everyone here. I can see all kinds of answers here. Anyway, we are given if a plant bearing pure axial flowers, right? Pure axial flowers would be capital A, capital A. A plant bearing pure terminal flowers, that would be small a, small a. What is the F1 generation's phenotype and genotype? F1 generation is this. It would be AA. What is its phenotype? It would be axial. There is at least one capital A. So this is axial. Phenotype will be axial. And genotype will be heterozygous. Okay? Yes, phenotype is all axial flowers. Genotype is all heterozygous. There is no ratio. All of them have the same genotype. So there is no 3:1. We are talking about F1 generation children. Everyone is just jumping directly into 3:1. Okay? So No, no. Okay, we are talking only about the F1 generation here. All right, and let us go to the next one. Draw a Punnett square board, okay, to show the gametes and offsprings when both the parent plants are heterozygous for axial flowers. Okay, when both the parents are heterozygous, what happens? Oops. Okay, this would be the case. Both the parents are heterozygous. Now we can talk about it using a Punnett square, because the question itself says draw a Punnett square board. How many will you have? You will need 4 boxes because it's a monohybrid cross, right? 4 different types can be seen. Now, the different gametes will be capital A, small a. Here also it's going to be a capital A and a small a. So these are the different gametes, and the combinations will be AA, heterozygous, here again heterozygous, and here it will be homozygous. Okay, very clear to everyone, right? This is very simple because it's monohybrid. Okay, yes. And the next question. What is the phenotypic ratio and genotypic ratio of the above cross shown in 2? So phenotypic and genotypic ratio here. How would you write that? Phenotypic and genotypic ratio of the F2 generation. This is basically showing you the F2 generation, right? Because F1 is this. From sub question 1 we saw F1 is that. Selfing of that has led to this. Okay, now it's coming to the answer that all of you were talking about. We have a 3:1 ratio for axial to terminal. What about genotype? 1:2:1. Yes, like Shagun and many others say, genotypic ratio would be 1:2:1. 1 capital A, 2 capital A, small a, and 1 small a, small a. Yes. So that brings us to the answer for question number 3 also. The next one is a very direct question. State Mendel's law of dominance. Of course, all of you would know this. What is Mendel's law of dominance? According to Mendel's law of dominance, when you have to state it, it has to be in the wordings that Mendel used. Mendel never used the term allele and gene. Those terms were discovered much later or proposed much later. Okay, factors is what Mendel always used. Whether it's allele or gene, Mendel used the term factors. One member of the pair dominates the other. Okay, and the dominant one is what dominates, and the one that gets suppressed is called recessive. Okay, yes. All right, so let's go ahead to the last sub question for this one. Name 2 genetic disorders commonly seen in human males. Can someone tell me the answer to that? Yes. Very good. Again, Dranzer gigs the first. Hemophilia and color blindness are 2 common diseases, 2 common genetic disorders which are seen in males. Hemophilia and color blindness. Now, why do you think this is more common in males and not in females? Basically because this is carried by the X chromosome. The gene for these traits is carried by the X chromosome. And because of that, we know a male's combination is XY. When just one X chromosome is affected, there is no copy of the X chromosome to mask the effect of that one. And because of that, a male can never be a carrier for such diseases. He is either normal or affected. Whereas females, for them to be affected by hemophilia or color blindness, they need 2 copies. If they have one normal X chromosome and the other one which is affected with hemophilia, they will still be normal externally. It's only that genetically they are carrying the disease, which is why, as far as being affected is concerned, these diseases are more frequently seen in males than in females. Okay. All right, and that brings us to the end of this question. Now let us go to the next one, which is also from genetics. Find out the progeny of a marriage between a color-blind man and a carrier woman. Okay, what would be the genotype of a color-blind man? Who's going to tell me that? Color-blind man and a carrier woman. A woman who's carrier for color blindness. Yes. All right. Color-blind man's genotype will be XCY. Okay. The female, the carrier female's genotype would be XCX because she's a carrier, which means she has one X chromosome, which is normal. Okay. What is our question? You have to find the progeny of a marriage between them. How do you calculate this? Again, using a Punnett square. Okay, the gametes are XC, Y, XC and X. Okay, XC, XC. This will be XCY, this will be XCX and this will be XY. Okay, so now when you look at this Punnett square, okay, now when you look at this Punnett, let me draw it once again, that was my mistake. It's an XCY crossed with XCX, right? So the progeny would be XC and this is a normal X, right? So this will be XCXC, which will be a color-blind female because two copies of the affected X chromosome. This will be XCY, which will be an affected male because there is a Y chromosome and the existing X chromosome has a C. C is standing for color blindness. Okay? Yes. I have explained all this when I taught you the chapter. Okay? Then the next one is XCX, which is a carrier female. She has one normal chromosome and one affected X chromosome. And the next one would be XY, okay, which is a normal male. So when you have to explain the progeny, how would you do this? You can say among the progeny, of course you can see these are the girls, these are the boys. The girls, the females are, this is the icon for female. Females are all XCXC or XCX, which means females are either color blind, 50% of the females are color blind and 50% of them are carriers. Okay, now just considering the females, this would be your ratio. 50% color blind because they have two XC chromosomes, 50% carriers because they have one normal X chromosome. Okay? Yes. All right. And then what about the males? Males icon is this. Among the male children, there would be 50% who are color blind and 50%. So in case you have more confusions on questions in genetics like these, please go back to my older videos where I have discussed a lot of such problems. Do go back and have a look at them. Things will be much clearer for you. Okay. And now coming to the last question for today, which is from the chapter Absorption by Roots. Okay, so the diagram given here represents a plant cell after being placed in a strong sugar solution. You are supposed to study the diagram and answer the questions. Okay, now what is the state of the cells shown in the diagram? You must know what I'm asking about. What is the state of the cells? Flaccid cell, very good. Again, Ranzer Gix the first has given the answer. It's not plasmolysis. State of the cell. Jay, we are not talking about a phenomenon. We are talking about a state of the cell. It is a flaccid cell. Okay, so this is how you would spell it. Flaccid. It's a flaccid cell. Now, just in case you do not remember that, I've put it here for your reference. This is what a turgid cell is. When you place it in a dilute solution, it becomes turgid. This is when you place it in an isotonic solution. Flaccid is when you place it in a slightly concentrated solution, and plasmolized is when you place it in a highly concentrated solution, wherein the cell membrane moves shrinks away from the cell wall. Okay. Yes, so that is about the difference between turgid, flaccid and plasmolized. How do you know it's flaccid and not plasmolized? Basically because, have a look at this, the cell membrane is still intact. It's not shrunk inside, but it is still a lot away from the cell wall. It's not too much shrunk inside and it still has some shape to it within the cell membrane. That's why we know it's flaccid and not plasmolized. Okay, yes. And the next question, name the structure that acts as a selectively permeable membrane. Yes, very, very, very simple. Basic question, name the structure of the cell which acts as the semi-permeable membrane. Cell wall, no. Plasma membrane, yes Shagun. Plasma membrane acts as a selectively permeable membrane. Exactly, so that is your answer here, plasma membrane. The next one, label the parts numbered 1 to 4. Now, in case you cannot see the labels, let me once again say this is 1, okay, this is 1, this is 2, okay, this is 3, and this is 4. What is 1, 2, 3, and 4? What is 1? 1 is, no, 1 is not cell membrane. 1 is, okay, let's look at it. Let's label it here. 1 is the cell wall. As you can see, 1 is the cell wall, the outermost, right? So it is the cell wall, this part. 2, what is 2? This part, inside the cell wall, what do you see? Yes. This strong sugar solution, exactly as Shagun says, sugar solution is 2. 3 is the cell membrane, we've already discussed, that is why we identified it as a flaccid cell. And 4 is the nucleus. Okay, yes, and answering that much fetches you full marks. And finally, not finally, I think there is one more after this. How can the above diagram be brought back to its original condition? Mention the scientific term for the recovery of the cell. Okay, yes, tell me. How can you bring it back to its original condition? How can you make a flaccid cell back to a normal cell? That is by placing it in a dilute solution or a hypotonic solution. Okay, you call that phenomenon deplasmolysis. Now this is plasmolysis, wherein when you put it in a highly concentrated solution, it shrinks, the contents shrink from the cell wall and it comes back to the normal shape. Okay. Yes, by keeping it in a hypotonic solution. That's fine. Hypotonic is fine. It needn't necessarily be pure water. It can be any dilute solution. Okay, because what we earlier had was highly concentrated. Okay, now one more last question. That brings us to the end of today's class. State any 2 features of this plant cell which is not present in an animal cell. This is extremely simple. Basically asking you the difference between plant cell and animal cell in a fancy way. Okay. Yes, this you can say any of these. In an animal cell, a cell wall is absent, whereas in this plant cell there is a cell wall. Animal cells don't have a chloroplast, whereas plant cells have a chloroplast. Or you can say anything. Vacuoles are much bigger, but then I think specifically the question is which is not present in animal cell and present only in plant cell. So it has to be the cell wall and chloroplast. Okay. Yes, large vacuole, that's what I was saying. A better answer would be cell wall. Okay, yes. All right, and that brings us to the end of today's session, children. Let me give you a homework question. All of you have a look at it. It looks slightly long, but it is actually very interesting. In order to study the process of osmosis, a student has taken 3 potato cubes, put them in 3 beakers containing 3 different solutions. Okay, and after 24 hours, in the first beaker, the potato cube increased in size. In the second one, it decreased in size. In the third one, there was no change. Okay, why is it that in the third one, there was no change in the size of the cube. You have to explain this to me. Think of it with reference to osmosis, all this tonicity, hypertonic, hypotonic, and all of that you have learned. Think of it with that sense, in that sense, and tell me the answer to this. Why is it that there was no change in the third beaker? Okay. Yes. And once again, children, if you have enjoyed today's session, do remember to hit the like button right now. Share it with all your friends, especially your ICSE Class 10 friends. And subscribe to the channel if you haven't done it yet, because we still have a lot more amazing content in store for you in the coming days. As I told you, in the next 4 sessions will also be diagram series. We will be taking 2 or 3 chapters at a time. Okay, so do remember to join me. But in the meantime, any questions that you have, do send me an email at ambika.gopalakrishnan@vedantu.com. Okay, children, thanks a lot once again for being so active. I would love to have your participation again in the next session also. So until we meet again, this is Ambika signing off. Bye-bye.